Hello and welcome to video 2 for week 7, talking about cellular respiration. So in the previous video, we went through aerobic respiration and how glycolysis is the start, converting glucose into pyruvate in the cytosol, which then transitions into acetyl-CoA and the Krebs cycle, producing those electron carriers. And in the electron transport chain uses oxygen to produce ATP. Now, we talked about what happens with oxygen, but what happens when you don't have oxygen? So for just a very short amount of time, just for a few seconds, our body actually has a pathway that's an example of substrate level phosphorylation, where a phosphate group is directly added back to ADP. And this works just for a few seconds with how much phosphocreatine we have in our bodies. So some of you bodybuilders might be familiar with this molecule because some people take it as a supplement, which I don't recommend. But that's what it's doing. It's providing that phosphate group to directly form some ATP. But this only works for a brief moment. So what happens when you're out of oxygen for longer? So we can now talk about fermentation. So fermentation is what happens when you are not doing aerobic respiration. Aerobic respiration requires the use of oxygen and occurs in the mitochondria. Anaerobic respiration is going to start in the cytoplasm and it's going to end in the cytoplasm because no oxygen is going to be involved, no electron transport chain in the mitochondria. So all the energy that's going to get released in these pathways we're going to see is just the energy from glycolysis. So fermentation uses substrate level phosphorylation, that direct adding of a phosphate group to ADP to make ATP. And again, it's because it's what's happening in glycolysis. So you already saw the process of glycolysis with that initial steps that require 2 ATP and then the output where you get 4 out. But in doing so, you get that electron taxicab of NADH. And normally, if you were doing aerobic respiration, that's good. That's what we need for that 4th and final step. In fermentation, that's all you do. You just do glycolysis. So in order for glucose to continually being broken down into pyruvate to get those 2 ATP out, we need to keep regenerating NAD+. Now, different organisms do this in different ways. Bacteria and yeast do this in a process called alcohol fermentation, where that pyruvate gets ultimately oxidized to carbon dioxide and it produces acetaldehyde, which oxidizes NADH back to NAD+, and in doing so produces ethanol. You might be familiar with ethanol as it's what we drink in alcohol as beer. This process of alcohol fermentation also is going to cleave that pyruvate from a 3-carbon sugar into acetaldehyde, which is 2 carbons, and in doing so produces carbon dioxide. This is actually what we see in breads when you're seeing those air bubbles. That's that release of carbon dioxide from the yeast that's eating all the sugar. So alcohol fermentation is one of 2 ways we're learning about that you can regenerate that NAD+ to keep glycolysis going and to keep producing the little bit of ATP. And one of these interesting things about this oxidation of pyruvate into alcohol is it actually can happen in human bodies too. So it's not by our own cells, we don't have these enzymes, but sometimes a person can be infected with a particular yeast that is doing alcohol fermentation and they can actually get drunk just by eating sugar. So again, alcohol fermentation, you are taking glycolysis and you are producing that NAD+ back in order to keep the whole cycle going. And in doing so, you convert pyruvate into ethanol. So we humans do not do this in our own cells. But what we do is lactate fermentation. This is also what other bacteria do and some other yeast and fungi as well. But this is, again, the purpose of it is fermentation. So you need to regenerate NAD+, but without oxygen, without doing the electron transport chain. And in this case, that 3-carbon pyruvate gets converted into a 3-carbon lactate. And in doing so, regenerates NAD+. You might also be familiar with this because this is what the fermentation that is happening in cheese and yogurt to make it delicious. And when I said humans do this, we normally want to be using glycolysis as the step 1 in aerobic respiration. But when we don't have enough oxygen, that's when our muscle cells will do this. So again, you want to understand anaerobic respiration. In animal muscles, we are regenerating that NAD+, and in this case, we're just converting pyruvate to lactate. So again, as an overview or a summary of how ATP is produced, when there is oxygen present, glycolysis and the transition and the Krebs cycle and the electron transport chain and chemiosmosis is going to produce the most ATP and produce carbon dioxide and water as a result. You can also make ATP from that creatine phosphate. And then there's both of those anaerobic pathways where oxygen is not present. And in humans, we can make lactic acid, or in yeast, they can make alcohol. So all of this was starting with glucose. And as we can remember, that makes sense. We talked about how plants store glucose as starch, how we eat that starch, break it down, and when we have extra, we can short-term storage in glycogen. We can break down that glycogen back into those glucose molecules, so we have the enzymes to cleave these glucoses. But we can also get energy from eating fats. So we spent a lot of time talking about triglycerides, which actually store more energy than carbohydrates. So when our blood glucose levels fall, our bodies can use the storage from triglycerides and to break down the triglycerides back into that glycerol and those fatty acids, and then ultimately break down each of those fatty acids and releasing the carbons as carbon dioxide. Proteins we can also eat and get energy from. In fact, you can nicely see here the amino acid alanine. Cleave off that amino group, you pretty much are already at pyruvate. So we can think about the different types of food we eat. They're going to go into aerobic respiration at different points depending on their compounds, and you can see how those molecules look very similar. Again, that amino acids are almost already at pyruvate if you remove that amino group. So you can see how the different components flow into the aerobic respiration. Now, lastly, we can talk about a little bit of regulation of this process. We talked about those 4 steps, starting with glycolysis, then going to the transition of the formation of acetyl-CoA and the citric acid cycle, producing those electron carriers, which are bringing them to the inner mitochondrial membrane and for oxidative phosphorylation. Now, one of those enzymes I pointed out at the very beginning of the first part 1 presentation was phosphofructokinase. Now, back as those examples of feedback inhibition, when our body has a lot of energy, we have a lot of ATP. ATP itself will go back and inhibit phosphofructokinase. Additionally, components in the citric acid cycle will go back and inhibit phosphofructokinase. So when we have plenty of energy, we'll get those signals so that our bodies will not follow through with glycolysis and oxidative phosphorylation. So to sum, remember we have all the different metabolic pathways, and metabolic pathways are meaning specific enzymes cleave one specific substrate, producing a specific product. And then another enzyme comes and takes that shape to cleave that reactant into a new product. So metabolism is the sum of all of these different reactions that break things down and build things up. Good luck.